Hello friends, welcome to my channel Miracle Agri Botany. Myself Dr. Snehal Tambe. I am working as assistant professor of agriculture botany at Kekewa College of Agribusiness Management, Nasik. Today's topic is Mendel's law of inheritance. In that, today we discuss different genetical terminology related to Mendel's laws of inheritance. In that, first terminology is gene. Gene is a small fragment of DNA and a heredity determined biological factor of an organism. And the term gene was coined by Johansson in 1909. The next terminology is genome. The total genetic constitution or composition of an organism is known as genome, and the organism complete set of DNA sequence is also known as genome. Then what is mean by allele? Allele is nothing but the alternative form of gene and which is present or occupy on the identical loci of the homologous chromosome. It's called as allele or allelomorph. And the term allele was coined by Batson and Saunders in 1902. Then next terminology is locus. What is meant by locus? Each gene occupies specific position on a chromosome is known as gene locus or loci. Then next one is dominant allele. What is meant by dominant allele? The allele that expresses itself in the same form, whether present in the homozygous or in the heterozygous condition, and it is represented capital letter T, for example, tall plant as a capital T. Then, what is meant by recessive allele? The allele which does not expresses itself when present with a dominant allele means when they are present with dominant gene or dominant allele, they suppresses their self is known as recessive allele. It expresses itself only in the homozygous form, and it represented by small letter. For example, for dwarfness is small t. Next terminology is phenotype. What is meant by phenotype? The actual characteristics appearance of an individual or physical appearance of an individual is known as phenotype. For example, color or shape or height of the plant. Then what is meant by genotype? The genetic constitution of an individual is known as genotype, such as capital R small r or small r small r. Then what is meant by homozygous? Individuals having similar alleles or gene is known as homozygous. Means either capital T or small t. They produces only one type of gametes. Homozygous produces only one type of gamete, either capital T or small t. And capital T and capital T are the two identical alleles for tallness. Similarly, for dwarfness, small t and small t are two identical alleles are produces from homozygous. Then what is meant by heterozygous? A heterozygous individual have dissimilar alleles for particular phenotypic traits on both the gene loci, and capital T for tall and small t for dwarf. Then what is meant by contrasting characters? Next terminology is contrasting characters. The individual plants features with marked phenotypic difference is known as contrasting characters, or it is also known as traits. For example, red flower color or white flower color, or next example is yellow and green seed coat color, and plant height as a tall plant or a dwarf plant. Then next terminology is F1 generation. It is also known as F1 indicates first filial generation. The progeny of a cross between two inbred lines is known as F1 generation. Then F2 generation, which indicates second filial generation. The progeny of F1 plants obtained by selfing. Next genetic terminology is inbred lines. The homozygous breeding line, which is obtained through self-pollination, is known as inbred lines. Then next, haploid. What is meant by haploid? The gametic chromosome number of a species is known as haploid and which is denoted by small n. Then diploid. The somatic chromosome number of a species is known as diploid and which is denoted by 2 small n. Then next terminology is gamete. A haploid cell, a haploid sex cell in plants and animal, for example egg or sperms, is known as gamete. Then zygote. Diploid cell resulting from the union of two gametes in sexual reproduction is known as zygote. Then pure line. What is meant by pure line? A plant which produces only one type of offspring or progeny from self-pollination is known as pure line. Then crossing. Artificial pollination between plants having contrasting genes or alleles for a character is known as crossing. And the progeny obtained from such crosses is known as a hybrid or a F1 generation. And from these progeny or hybrid, there are three crosses are involved in that hybrid or F1 generation. In that, first one monohybrid cross, second cross dihybrid cross, and third cross we obtain is the polyhydric cross. Then monohybrid cross, a cross involving one gene pair affecting one character is known as monohybrid cross, and such crosses obtained in phenotypic ratio is 3 is to 1, and genotypic ratio obtained from monohybrid cross is 1 is to 2 is to 1. Next cross is dihybrid cross, a cross involving two pairs of genes and each pair affecting a different character is known as dihybrid cross, and such a dihybrid cross give phenotypic ratio is 9 is to 3 is to 3 is to 1, and genotypic ratio obtained from dihybrid cross is a 1 is to 4 is to 6 is to 4 is to 1. Then next cross is polyhybrid cross, a cross involving three or more pairs of genes and each pair affecting a different character is known as polyhybrid cross. Then next terminology is direct cross. A cross between any two genotypes is known as direct cross. Then reciprocal cross, a cross between two genotypes in which the order of male and female parent is reverse, means change the position of either male or female parent is known as reciprocal cross. Then what is meant by back cross? Cross of F1 with either of its parents is known as F1 back cross. For example, red flower plants cross with white flower plants, and the F1 which get from or obtained from these two parents, we get F1 is all red plants. Here is the parents red flower plant and white flower plant, and gametes obtained from this red plant is capital R, and from white plant, gamete obtained is the small r, and the progeny F1 obtained from these two parents or these two plants, red into a red plant and white plant, we get all red plants in F1 generation. Then test cross. Cross of F1 with homozygous recessive parents is known as test cross. For example, crossing of F1 with white flower plant, because white is the recessive parents, because in F1 we get only red plants. So white flower or white flower plant is the recessive, are the recessive parents. So from these test cross, we get phenotypic ratio is 1:1 in test cross progeny. Whereas two gene difference we obtain it like 1:1:1:1 phenotypic ratio. Thank you so much for watching my channel.